regulatory person, I understand that this design and definitions are sometimes a little complex. I'll try to resolve or solve them or make them simple for you. And then, as Pranav had mentioned in his LinkedIn post, that we will then discuss about bioweavers as well. What are bioweavers? That we'll come to it after all these are covered. So let's ask these three questions to ourselves first. What is B? Why is B? And how it is done? Let's start with what is bioequivalence studies. Now, when you search on internet and in the guidelines, there will be several definitions given of bioequivalence studies or bioequivalence. And sometimes it is little confusing. So to make it little simple and make it practical and for you to understand it easily, I've broken down the definition of bioequivalence into 4 components, these 4 bullet points. Okay. If you focus on this, I think you will always be remembering what is actually a bioequivalence study. Whenever you have doubts, there is a Q&A tab in the webinar. You can raise your questions, and at the end of the webinar, we will try to resolve as many questions as possible. Okay. So bioequivalence definition has these 4 components. Let me explain you one by one. Bioequivalence study is a comparison between 2 products. Remember this 4th point. It's a comparison between 2 products, 2 pharmaceutical products. So what is being compared? The rate of absorption and extent of absorption is being compared. What is absorption? How the product from the GI tract, stomach or intestine, goes into bloodstream, and how much does it go? That is known as absorption. Now, absorption's rate and extent. What is rate of absorption? How fast the drug goes from gastrointestinal tract to bloodstream is the rate of absorption. You might have heard of the term Cmax. Okay, so Cmax is the rate of absorption. How fast? Whether in 15 minutes, 10 minutes, 1 hour, 2 hour. How fast does the drug go in the bloodstream? That is rate of absorption. Extent of absorption means for how long and in how much quantity does the drug remains in the bloodstream. Okay, you take the medicine, within 1 hour it reaches the bloodstream, but for how long does it stay in the blood? For 10 hours, 20 hours, 30 hours, 2 days, 1 day. So the amount of time and the amount of drug that stays in the blood is known as extent of absorption. And in statistics, we can evaluate extent of absorption through a term AUC, area under the curve. So rate of absorption, extent of absorption, both are compared between 2 products. And this Cmax and AUC should not be significantly different. Remember, this first point is most important. The definition of bioequivalence does not say that both products have to be similar. What it says is there should not be a significant difference. When you're comparing 2 human personalities, no 2 humans in the world, even the twins, are exactly the same. There must be some differences. So when you compare 2 personalities, you don't expect them to be same. What you expect is that the differences in their personalities and attitude should not be similar. Then you will call them similar personalities. Similarly, pharmaceutical products, you don't want them to behave exactly same CM, Cmax and AUC, but the differences should not be significant. So the definition of bioequivalence does not say that the rate and extent of absorption between 2 products has to be same. What it says is it should not be significantly different. So whenever somebody asks you the definition of bioequivalence, it's not necessary to of the textbook definition. If you know these four components, you will be able to explain in your own words that when two products rate and extent of absorption is not significantly different, they are called as bioequivalent. So this answers our first question. What is a bioequivalence study? For this question, if you have any queries, you can type in the Q&A box. Now coming to our second question. Why is bioequivalence required? Now, you know, whenever a new product is introduced, an innovator product is introduced, the company has done a lot of research over it. So to make profits, they keep the rate very high. The cost of the drug is very high, of innovator drug. And what happens after their exclusivity is over, after 8 years, 10 years, then generic players can enter. And the doctor can replace the innovator product with the generic product. But doctor will only have confidence in replacing the innovator product with a generic product when the doctor knows that both the products are bioequivalent. That means when I replace an innovator product with a test product or a generic product, its Cmax and AUC behaves similar to the innovator product. Then only it will give same efficacy and same safety. If blood concentrations vary, then the efficacy or safety may vary. If blood concentrations are less, then it may not give proper efficacy. If blood concentration of generics are high, then there may be safety concerns, toxicity issues. Okay. So why is a bioequivalence study required? To have confidence in generics that now the innovator can be completely replaced or majorly replaced. So this is why we are conducting a bioequivalence study. This is the why of our webinar. Now comes how is a BE study performed. So as I told you these terms, Cmax and AUC. So how it is performed? Human subjects. Bioequivalence study is kind of a clinical trial where human subjects are taken. And in most cases, healthy human subjects. Because we are not evaluating efficacy or safety. We don't want patients. We just want blood concentrations. So healthy human volunteers or healthy subjects are taken. Test product is given to them. The reference product is given to them. And from time to time, blood samples are withdrawn at 1 hour, 2 hour, 4 hours, till 24, 48 hours. And concentration versus time graph is plotted. This graph comes like this. Drug enters the blood. It slowly increases its concentration. At one point, it reaches its maximum concentration. That is known as Cmax. And the time when the Cmax is achieved is known as Tmax. Suppose it is achieved in 2 hours, maximum concentration. So 2 hours is the Tmax. And we follow these blood concentrations till the drug is completely eliminated from the body. Slowly declines, and at one stage, the concentrations become zero after 1 day or 2 days. And the entire time and extent when the drug remains in the bloodstream can be given by this AUC. This shaded graph is the AUC. That means how long does a drug stay in the body. So here you can see what is compared. Test product. This dotted line is for test product. And this green bold line is for the reference product. The Cmax of both and AUC of both, test and reference, is compared, and then it is finalized whether the drugs are bioequivalent or not. Okay. So this is the how of our webinar. How is BE study performed? Give to human subjects, measure the blood plasma concentrations, and evaluate Cmax and AUC statistically. Okay. So this was what, why, and how of BE. Now, this was just introduction part. Let's dig deeper into the designing of a BE study. BE study design. Some of you might already be working in regulatory affairs or other departments or in industry already. You must have seen this title of BE study. This is the most common title of BE study. A randomized open label single dose, 2-period, 2-sequence, 2-treatment crossover study to compare the bioavailability of test product and reference product in healthy adult human subjects under fasting conditions. This is the most common title. And as a beginner in clinical research in bioequivalence studies, I always had doubts what these all terms mean. These are not just definitions, but through these terminology, you will be exactly able to understand the BE study design. So what I want to do is explain you each of these terminologies and give you a proper definition of what is randomized open label single dose 2-period 2-sequence 2-treatment and crossover design. And once you understand these concepts, then later you will find bioequivalence studies very interesting. So one by one, we will try to understand this terminology. In the next slide, this is an important slide. If you want to capture a photo, you can capture. Through this slide, I will be explaining you what is randomized, what is 2-period, what is 2-sequence, and what is crossover. At least 4 terminologies will be learned through this slide itself. Okay, so this slide may take time, 5 or 10 minutes for you to understand. But this is the most important slide for you to understand. So first, let us understand what this 2-period is. As I said, BE study is done in human subjects. According to the guidelines of various countries, at least 12 human subjects should be evaluated, minimum 12 subjects. But usually the number of subjects is 24, 30, 36, 28, 40. And sometimes they can be as high as 80 subjects, 100 subjects in rare cases. But usually it is 24 to 36. Here I have taken example of the minimum criteria, 12 subjects, for you to easily understand all these concepts. So consider that there are 12 human subjects in the BE study. So all these human subjects will take 2 treatments. Once test product in period 1 and in another period reference product, or reference product in the first period and test product in the second period. So the BE study is done in 2 periods mostly. First period, 1 treatment, second period, another treatment. That's why they call it 2-period. Okay. There can be 3-period studies also, 4-period studies also, but we won't go as deep as that. Once you learn this 2-period studies, it will be easier for you to understand 3-period and 4-period studies. But 80% of the BE studies, according to me, are 2-period studies. So all the human subjects in period 1 will receive 1 product. In period 2, they will receive another product. So this was 2-period. Now coming to crossover, another terminology is crossover design. What is crossover? Crossover means if the subject in period 1 took test product, second period they cannot take test product again. They have to take reference product. That means the treatments are crossed over. Okay. It's not that all the subjects in period 1 are taking test. Some subjects are taking test and test. Some subjects are taking reference and reference in both periods. They have to cross over the treatments. That is the crossover study. Apart from crossover study, there is a parallel study as well. which we will not go through now because it is not common. Okay. So crossover study means if in the period 1 test product was taken, period 2, it will be reference. If it was reference, then it will be test product. So this is crossover study. Another term is two treatment study. There are two treatments. Same subject is receiving test product also, reference product also. It's not that we are giving only test product to all subjects. And in another bio study, we will give, in a separate study, we will give a reference product. No. In the same study, two treatments have to be given, test and reference both. That's why the definition says two treatment. So I hope now you are aware about what is two period, two treatment, crossover study. Okay. Now coming to another terminology, randomized. Randomized. Before we learn randomized, let's learn two sequence. What is this two sequence? We saw what is two period. We saw what is two treatment. We saw what is crossover. Now let's understand what is two sequence. You can see subject 1 is receiving test product first, reference product first. So the sequence for subject 1 is TR. For another subject, it is RT, first reference in period 2 test product. TR is one sequence, RT is another sequence. You can see all the subjects are having either of these two sequences, either TR or RT. TR, RT, RT, TR, TR. No subject is having TT, RR. Okay, those sequences are also possible, TT, RR. They are having only two sequences, either TR or RT. So this is known as two sequence study. So this gives us the definition of two period, two treatment, two sequence. Okay, two treatment can be three sequence also. As I said, TR, RT, RR, TT, four sequences. But guidance says that if you are using two treatments, there should be only two sequences, TR and RT. No subject should be repeated the treatment. That means in period 1, if they are receiving test, again, test product is not allowed. So TR and RT, these are the two sequences. So this gives us the definition of two sequence, two period, two treatment, crossover study. Now come to the first definition, randomization. See, these sequences TR and RT are random. It's not that we are giving alternate TR, RT, TR, RT. Okay. So this is known as TR randomization of the study. And this randomization is done through statistical software. Human being does not decide whether this sequence should be TR, RT, TR, RT, RT to avoid bias. Okay, this randomization schedule is decided by the software, the statistical SAS software. So this is randomization of the sequences. So the sequences are randomized. Why this crossover and randomization and two sequences are required? This is required to avoid bias. Okay, if there is no randomization, then investigator of the study, that means the head of the study, clinical department, can choose that to this particular patient, this particular human subject looks healthy and young. I will give test product to him to show good blood levels. And little older or weak or middle-aged people or obese people, I will give reference product to show less concentrations. So investigator may do some bias or give choice of the drugs according to his selectivity. To avoid this bias, crossover and randomized designs are done. So I hope the whole design these 5 definitions are clear to you. Once again, I will quickly explain what are all these things. So two treatment means test and reference products are given. Two period means in different periods. In one period, one treatment. In second period, second treatment. Crossover means if the subject received test product in first period, then they have to receive the reference product in second period. That is crossover. Randomized means these two sequences are randomly given to the subjects, not in a fixed sequence. So to avoid bias. So with this slide, we now understand what is two period, two sequence, two treatment, crossover, randomized design. In EU-BE guideline also, you will find that ideally you should use this design, randomized, two period, two sequence, two treatment, crossover. And some exceptions are given, that in which exceptions you can choose parallel design, three period design. Okay, you can go through those exceptions. Now, as you are aware about all these definitions, now it will be easier for you to understand. Now, in this study design, two definitions still remain, open label and single dose. Let's jump to those two definitions now. What is open label study? See, there is another study type, blinded study. So normally in a clinical study, three parties are involved. The human subject who's taking the drug, investigator who is doing the study, and the bioanalyst who is doing the analysis on LCMS of the blood samples. So ideally these three parties are involved majorly. Many other roles are there, QA, pharmacist. But as far as this open label and blinded is concerned, these three parties are the main roles which are concerned. So in an open label design, the subject knows the product, whether I'm receiving test product or reference product in period 1 and period 2. Subject is aware. Investigator is also aware that whether test product is being given or a blinded or a reference product is being given to the subject. But the bioanalyst does not know whether, even in the open label study, bioanalyst does not know whether he is analyzing test product in the sample or reference product in the sample. Only the sample ID will be provided to him. That's sample ID XYZ235. You have to analyze. Whether it is test product, reference product, he does not know. Now, why he is not allowed, that I'll explain shortly. What is a blinded study? In blinded study, subject also does not know which treatment he is receiving, reference product or test product. Even investigator does not know. They only know the sample ID. But it is test product or reference product, they do not know. The bioanalyst obviously does not know, because in open label study also, bioanalyst does not know whether they are receiving test product to analyze or reference product to analyze. Now, what is the purpose of doing a blinded study, and why a bioanalyst is not known, and why in BE studies, blinded study is not required? That I'm going to explain. You must have heard of the term placebo effect. Let me give you an example. If you have been suffering from cough for last 15 days and you have taken allopathic medicines and codeine or fall codeine or whatever, and it is usually happening that we have cough many times which doesn't go away for 15 days, 30 days even, sometimes in winters or any other season. And some relative of you comes to you and says, take this Ayurvedic medicine, and it relieved my cough last year, and it has relieved cough of hundreds of people where allopathic medicines have been ineffective. So even if that Ayurvedic product does not have proper efficacy, the trust that he brings to you, that it has removed the cough of or treated the cough of hundreds of people, it's a magic medicine. you will feel 30-40% efficacy with that trust itself. Okay, that belief. So whenever there is efficacy, or somebody, if somebody uh, gives you a fear that if you take this medicine, you will have such side effects, safety issues, then that fear in the mind also makes an actual clinical impact. There are more chances that you will suffer from those side effects. So efficacy and safety always have this placebo effect. To overcome this placebo effect, many clinical trials are blinded. Subject does not know that they are taking innovator product or test product. If they know that they are taking innovator product, they will have confidence and trust that this is a well-known product. It will give me a good efficacy, and it will give a placebo effect. Okay. But if they do not know whether they are taking innovator product or test product, then that placebo effect will be removed. Okay. So the actual efficacy of the drug or safety of the drug will be known through the clinical trial. But why in this bioequivalence study we are not doing blinded study? Because the bioequivalence study's purpose is to measure blood concentrations, not efficacy or safety. Even if I know I'm a human subject taking part in bioequivalence study, suppose, and I'm taking reference product, even if I know that reference product is a well-known product, innovator company, good quality, still it will not impact my blood concentrations because blood concentrations are not efficacy and safety, they are pharmacokinetics, and pharmacokinetics parameters are not impacted by placebo effect. That's why bioequivalence studies do not require blinding. Okay, let the subject know what they are taking, let the investigator also know what he is giving, but the bioanalyst should not know. Why in an open label bioanalyst is kept blinded? Because if the bioanalyst knows, he can do some bias during analysis or some manipulation during analysis to give good results of the test product. Okay. So in any case, bioanalyst is not known or not told what he is testing. Okay. So to avoid this manipulation or this issue, bioanalyst is blinded in an open study, open label study as well. And blinding study is done majorly for efficacy safety trials, phase 3, phase 2 trials, not for bioequivalence studies. Because bioequivalence studies, we are measuring blood concentrations, and blood concentrations do not have placebo effect. So this is open label and blinded study, and all the guidances says that open label B studies are okay. But Health Canada says B study should also be blinded. So if you go through Health Canada's guidance, although it does not have any placebo effect, still Health Canada recommends doing a blinded study. Okay, all other authorities, US, EU, Australia, or any other authorities accept open label studies. If you have any queries regarding this open label and blinded studies, you can type. Going to the next terminology in the bio study design, single dose. We are measuring blood concentrations. So we do not need multiple doses. A single dose of test product and a single dose of reference product will give us enough information about the blood concentrations. And we are doing it in healthy volunteers. It is not ethical also and safe also to give multiple doses to healthy volunteers. Okay. If single doses are giving blood concentrations, then we do not need to expose healthy volunteers to these treatments, which can cause safety concerns to them. So that's why a single dose study is always preferred. So now let's go back to this design of BE studies. I hope now it is clear what is randomized, open label, single dose, 2 period, 2 sequence, two treatment, crossover study. To compare the bioavailability of healthy human subjects we are taking healthy human subjects because we are just measuring efficacy or blood concentration, not the efficacy and safety. Fasting conditions, fasting or fed, that I'll come to it very shortly. Remaining of the design is now well, I hope is well understood to you. To the next portion of our webinar, designing a bioequivalence study. Okay. Now, if you want to take a 5-minute break, it's okay. You can take a 5-minute break. It's 11:37. Please come back at 11:41. We will start at 11:42. You may drink water or whatever. Relax. Yeah, welcome everyone. I hope you had a relaxing 5 minutes. Now go back to our topic. Now this was basic definitions and basic terminology and basic concepts that we learned in the first half. Now what we learn is designing a bioequivalence study. Now we are moving to the next step. If not only for the interviews, if you are asked to review a BE study protocol, a BE study report, or draft a protocol yourself, then you need to know some designing parameters, how BE studies designed. Okay. So how the protocol is designed, I will be giving you some basic concepts about them. We will learn these 7 parameters or 7 points, how to choose the reference product, how to choose the strength, whether to conduct a fasting or fat study, how to calculate the sample size. That means how many subjects, 12 subjects, 24 human subjects. What is washout period and how it is calculated. What should be the sampling schedule? At what time points we need to withdraw the blood from human subjects? Housing conditions. How long will the human subject stay in the BE center? These 7 parameters we will try to understand. Then there are several other parameters as well, but they are not as important as these 7. So for a regulatory affairs professional, the most important point to learn in BE studies, even if you do not know other aspects of the BE designs, you must know this aspect, how to select the reference product. Even at a higher position, when you have 10, 15, 20 years experience, again and again you will be asked this question, how do you select the reference product? Okay, so pay special attention to this slide. Reference product selection. If you are preparing a dossier for different countries like US, EU, Australia, New Zealand, Canada, or UK, in your BE study, what should be the reference product that we will be covering in this slide? Suppose your dossier is for US market and you are conducting a BE study for that dossier. Then the reference product, test product is manufactured from your company. So there is no doubt about the test product. It's your company's product. But the reference product, the innovator product must be procured from US. Forgot to write the, because US is called the US. Okay, jokes apart, must be from the US. It should be procured from the US itself. You cannot procure an innovator product. Suppose atorvastatin innovator product is Lipitor. So you cannot just take Pfizer's Lipitor from any country, Europe, Australia, India. That Lipitor reference product must be procured from US. It should be made for US. Okay. Similarly, in EU, if you're targeting an EU dossier, in your BE study, you must have the reference product from EU. From any country of the EU, if you are targeting, there are 28 countries or more than 28 countries in EU. So the reference product can be procured from any country, Germany, France, Croatia, Spain, Sweden, wherever. But it should be an EU country. So if you are targeting one product for US and EU, that means you have to have 2 different BE studies. In one BE study, US reference product, in another BE study, EU reference product. So 2 BE studies have to be conducted separately. Same BE study won't suffice. While in all these countries, most of them allow using EU or US reference product or EU or US BE study. But not in every case. In some cases, they will allow. Australia allows using the reference product from US and EU if you follow certain criteria. New Zealand also allows using a US, Australia, or EU reference product BE study if you follow certain criteria. The main criteria is the innovator company in both the countries should be same. Suppose in Australia you are using US reference product or US BE. The innovator in US, suppose Pfizer, should be the innovator in Australia as well, Pfizer. Lipitor from Pfizer in US is acceptable as a replacement of Lipitor in Pfizer or Pfizer in Australia. So main one is this criteria. The company should be same. Then you have to match some uh, dissolutions and qualitative, quantitative compositions apart from this. But the first thing is the company should be same. If the company is same, their manufacturing method, composition, all would be same. This is assumed by the regulatory authorities. So the reference product for Australia should be from Australia, but can be from US and EU if you follow certain criteria. Similarly for New Zealand, similarly for Canada, US and EU BE study would be acceptable, mostly in immediate release dosage forms. You have to follow the guidelines of Canada. Here for New Zealand, you have to follow New Zealand guideline. Here you have to follow Australian guideline. UK also accepts UK, US and EU, and sometimes Australia reference product also. Preferably it should be from UK, but if not, then US, EU also is acceptable if you prove that UK reference product is equivalent to US or EU reference product. Okay, so this is the most important thing. How will you search the reference product? For US, you will search in Orange Book. There is a separate column of RLD. RLD and RS, you can search the reference product there. From EU, you can search on MRI index or public assessment reports. In Australia, you can search on EBS portal. New Zealand also have their own database of reference products. In Canada, there is drug product database. In drug product database, you have to fulfill certain criteria to understand which one is the reference product. And UK also have their separate database where reference products are mentioned. So in individual database, you have to check which is the reference product. Okay, so this is the first criteria of designing a BE study, reference product selection. Coming to the next topic, which is also important, as important as reference product selection. Strength selection. Suppose a product has 3 strengths: metformin tablets 500 mg, 850 mg, and 1000 mg. Do we need to perform BE study on all these strengths? No. Generally, BE study is performed on the highest strength. Why highest strength? Because highest strength will have the highest variability when it goes in the body, in the bloodstream. High concentrations, high variability. Okay. But to get waiver means permission of not doing BE study on these 2 strengths, biowaiver. To get biowaiver of these 2 strengths, the lower strengths, you need to fulfill certain criteria. These are the 5 criteria that you need to fulfill according to the EU BE guideline. This lower strengths, 500 and 850 mg, should have the same manufacturing process as the 1000 mg. They should have the same qualitative composition. All the strengths should be quantitatively proportional. And you have to prove that their comparative dissolution profiles are matching. 1000 versus 850 and 1000 versus 500 mg have comparative dissolution profiles. And then you have to prove one more thing, linear pharmacokinetics. Linear pharmacokinetic means that suppose AUC of 500 mg is 100 units. Just an assumption, 100 units. Then 1000 mg is double of 500 mg. The AUC of 1000 mg should be 200 units, double than 500 mg. Similarly, Cmax If Cmax is 10 units, for 1000 mg, it should be 20 units. 25% variation is allowed. For this, if it is 100 units, for this one, it can be 160 units or 240 units. 20% variation is allowed. So that much variation is allowed. How will you know that the AUC and Cmax are increasing proportionally? You have to search literature database. You have to search on literature whether anybody else priorly has done pharmacokinetic studies. Innovator must have done. So they will write that with increasing dose, Cmax and AUC increase proportionally. Not only in the literature articles, many a times this information is given in the label itself, sometimes in the SPC, product name graph, that with increasing doses, the AUC and Cmax increase proportionally. So this is one of the important criteria which you cannot do in lab. You have to search on literature that whether AUC and Cmax are increasing proportionally. If they are increasing proportionally, then you are allowed to do the BE study only on the highest strength, and you get BioWaiver for the lower strengths. So this is one kind of BioWaiver, strength-based BioWaiver. You don't have to do BE studies on all these strengths. These criteria are applicable to IR dosage forms, immediate release dosage forms. For modified release dosage forms, you may have to do multiple studies on lower strengths and higher strengths also. I recommend you go through modified release clinical trial guidance from EU. There it is clearly mentioned in which kind of modified release dosage form, how many strengths BE studies are required. Okay, this I have explained for immediate release dosage forms. So the second aspect of BE study designing is strength selection. Mostly you will select highest strength. If the pharmacokinetics is non-linear, then you may have to select 500 mg lowest strength as well. Further details are given in EU BE guide. But the most common situation is we choose the highest strength. Coming to the next aspect in the BE design itself, it is mentioned fasting study. So how do we know whether to conduct a fasting study or a fed study? Now let me give you a visualization of how a BE study is performed before going into fasting and fed studies. The subjects, human subjects, are screened before the study starts, are screened 21 days in advance, not more than 21 days. Suppose the study is going to start on 22nd June. So all the screening, the minor criteria, major criteria, inclusion criteria, everything is done 21 days in advance. That means either after 2nd June, between 21st June, they will be screened. On the study day, they will be called to stay in the CR. There will be beds. The human subjects will come. Their health checkup will be done. Physical examination will be done. And in the evening time, suppose today is Sunday, today in the evening time, they will be called in the CRO, which is kind of an hospital. There will be beds, ventilators, laboratory, everything. So subjects will be asked to sit on those beds. At 8 p.m., generally at 8 p.m., they will be served their dinner, or either 8 p.m. or they will be asked to finish their dinner by 8 p.m. Then they will be allowed to rest. Next day, 8 a.m. in the morning, that means Monday 8 a.m., they will be given, each of them will be given a single dose of drug with 240 ml of water in most cases. Before 8 a.m., that means 1 hour before, 7 a.m. to 9 a.m. or 10 a.m. For these 3 hours, they are not allowed to take anything, even water. They will only get water with the drug at 8 a.m. 240 ml. So For last night, 8 hours, 8 p.m. to morning 8 a.m. There is a gap of 12 hours. So the guideline recommends they should be fasted for at least 10 hours. So even if they finish their dinner at 10 p.m., still there it will fulfill the criteria. So at least 10 hours fasting is required in a fasting study. But then there are some fed studies as well, where after dinner in the previous night, in morning 7 a.m. they will be served high fat, high calorie breakfast. So all studies, most studies are fasting studies, but then there are some fat studies as well, where high calorie and high fat, high fat breakfast is served. Then after 1 hour or so, they will be given the test drug or reference drug. So how do we decide whether to do a fasting study or a fat study? It depends on the labeling. If the innovator product label mentions that the drug has to be taken on empty stomach, then you must perform the BE study on empty stomach fasting. If the SPC or the labeling mentions take the drug with food or meal, then you have to do a fed study, where in the 7 a.m. you will serve high calorie, high fat breakfast. If the labeling does not mention anything, empty stomach or with food or meal, if nothing is mentioned, then you have to do fasting study by default. So fasting is the preferable study. But if the labeling mentions the drug has to be taken on fat state, then you have to do a study with food or meal or a fat study. So how do you decide fasting or fat? Depends on the labeling. This is for EU, Australia and Canada and majority of the countries. US is little different. For US, previously the guideline mentioned that for all the products you have to do fasting and fat study both to see the variability. But now US FDA has started giving product specific guidances. If you search on Google, suppose your drug is metformin, you will search metformin US FDA product specific guidance. 2-page PDF will open. In that it will be mentioned to do fasting study and fat study or do any one of them. So you have to follow US FDA product specific guidance. If there is no product specific guidance for your product, you have to do both fasting and fat. So US requires double investment, 2 studies, even for the immediate release dosage forms. And this is also for immediate release dosage forms. Okay, so this is how you will decide whether to do a fasting study or a fat study. To a little complex, not very necessary for a regulatory person, but it's a good to know topic. How is the sample size calculated? What is sample size? Means number of human subjects that you will enroll. As I said, the guidance says minimum 12 subjects should be there in the BE study. But usually the number of subjects are 24, 36 or something like that, between 20 to 30. So how do we decide how many subjects? Okay, companies always try to keep as less subjects as possible. Because let me give you a hint of how much the BE study costs. More the number of subjects, the CRO will charge you more. Previously, 8 to 10 years ago, it was a simple rule that one subject means 1 lakh rupees. If you're doing study on 24 subjects, we will charge you 24 lakh rupees. If 30 subjects, then so the companies prefer always using less number of subjects. But then they have to comply with the guidance. They cannot always use 12 subjects. So how it is decided whether to do 25 subjects? Nowadays, the cost has increased. This is 8 to 10 years ago cost, 1 lakh per subject. It's just a rough estimate. It's not the exact calculation, financial calculation of the BE study. It's a rough calculation. It can vary from nowadays, it may be 1.5 lakhs per subject. If you are doing a 24 subject study, it may cost you $36,000 or 35 lakh rupees or whatever. So always companies try to minimize the numbers of subjects, but they have to comply with the guidance. How to comply with the guidance? The sample size has to be calculated based on drug variability. Now, what is drug variability? All the drugs have some intrinsic variability. Suppose today I take a metformin tablet, 1000 mg myself, and the blood concentrations measured are 100 units, for example. After 4 days, I take the same drug, same me, same drug, same company, same formulation, same condition, staying in India in heat of 40 degrees Celsius, same health, nothing else, same weight. But if I take the same drug on Thursday, the blood concentrations may be 80 units, 90 units, not 100, 110 units, 120. So all the drugs and the genetics, my body also will react differently on different days. And the drug itself also has some intrinsic variability. So within the same subject also, within myself also, it is showing different concentrations. That property is known as intra-subject variability, or in statistical terms, it is known as intra-subject percent coefficient of variance. Okay. So if the intra-subject variability is high, you have to keep high number of subjects. Suppose if variability is 30-35%, you need to keep 45 to 60 subjects. This calculation is done through a statistical software where you put the values of the variability and it will give you the number of subjects required. Okay. Nowadays, you can do it through AI tools also. You ask any cloud, Gemini or ChatGPT that this is my variability. What should be the number of subjects required? It will give you the exact number or a tentative number. So it depends on intra-subject variability. Now the question comes, why for high variability drugs, high number of subjects is required? And why for low variability, we can choose 12 subjects also, which is the minimum criteria. So this is an approximate range. For moderate variability, you will need 24 to 36. And high variability, you will need 45 to 60 subjects. So for more than 30%, you will need 60 plus subjects or a replicate design. Okay. Not a crossover design, replicate design, 4-period design that you can learn through guidance. Okay, so why for high variability, high number of subjects are required? Now, before we go into that, let me explain you how do we know the percentage variability. So percentage variability is known through literature. You search various pharmacokinetic trials conducted in the past. There, this percent CV value is given for Cmax and AUC. That within subject also, this has this much variability. So you take 2-3 reference articles, you finalize the variability found, and then you finalize the number of subjects. And depending on that, your cost and budget and your designing will rely. Okay, so first question is how sample size is calculated through drug variability. Second question is what is drug variability? That means subject to subject or day to day it will change. Third question is how we know the drug variability through literature. Fourth question is why high variable drugs require more number of subjects. So because we are a cricket loving country, I have given a cricket analogy to understand this. If you want to compare Vaibhav Suryavanshi with Sachin Tendulkar, whether he is also young, 15 years. Sachin also started at 15 years. Okay. Whether he is as good as him or not, how do you compare? So batting in cricket is very variable in nature. Sometimes you score 0, sometimes you score 200. Sometimes you score 5 runs, sometimes you score 50. So with 1 or 2 innings or with 2-3 years, you cannot say that Vaibhav Suryavanshi is going to be as good as Sachin Tendulkar. How will you know? Because batting is a very highly variable skill. You need to see more number of innings throughout the years, 5 years, 6 years, 7 years. Then you will be able to say now he is one of the great cricketers. So because batting is a high variable skill, you need to compare across several innings and several years. Similarly, if drugs are also highly variable, you have to compare in several subjects. Then only you will get a good average. That yes, test product is as good as reference product. Okay. So that's why high variability of drugs require more number of subjects to understand whether they are equal or not. Okay. So this is how the sample size is calculated. Although all regulatory professionals are not required to know this, but it's always great to know more, so that when you are in discussions or doing a project or interviews, you know, and at some stage you will be given a task to manage finances as well. That this B study is costing 35 lakh. Why it is costing 35 lakhs? Then if you know this knowledge, then you will be able to say that this CRO is charging more. Let me ask another CRO, because the subject size is 22 only. Why the CRO is taking 30 subjects? Okay. So at some stage, this is definitely going to be helpful. Now you might have heard of this washout period many times. What is washout period? As I said, a BE study is mostly 2-period study. In first period, you are giving test product, for example. Then second period, you will give reference product. You need to allow the body to eliminate the first period drug completely. Then only you will start second period. Otherwise, the blood concentrations will get mixed up. So suppose on Monday you gave the drug, test drug, you cannot start period 2 on Wednesday or Thursday or any random day, Tuesday. Period 2 has to wait until the period 1 drug is completely eliminated. That complete elimination is known as the washout period. Now, how that washout period is decided? That is decided based on the half-life. You all have learned the definition of half-life. The time required for a drug to eliminate 50% from the body is known as half-life. So in the first half-life, 50% drug gets eliminated. Now 50% is remained. In the second half-life, that 50%, 50%, that means 25% will be eliminated. So after 2 half-lives, 75% drug is eliminated. After 3 half-lives, 87.5% drug is eliminated. 4 half-lives, 93.75. It is getting reduced by 50%. 50, 25, 12 and a half, 6.25, 3.12. After 5 half-lives, 97% almost drug is eliminated, only 3% remains. So EU authorities and other authorities in the guidance have mentioned that you should keep a gap of at least 5 half-lives. Start the second period, that is known as the washout period. Now take an example. If a drug has a half-life, you found out on literature and on labeling that the drug's half-life is 20 hours. Then you have to allow 5 half-lives, that means 100 hours to start the second period. That is around 4.5 days or 4.25 days. That means you are safe to start the second period after 5 days. So if you gave the first dose, test product dose on Monday, after 5 days, that means either Friday or Saturday. And to be on safer side, most CROs give a 7-day washout period in such cases, 1 or 2 days more. On next Monday, you can start another period. You can ask subjects to go home, come back again on next Sunday, we will start period 2. That is known as washout. period 1 with a gap of washout period. Then it starts period 2 to eliminate any carryover from period 1. Okay, so I hope this washout period calculation is clear to you. Now going to the next concept, which is also very interesting, sampling time points. As I said, the concentration versus time profile has to be done. Through this, we will be able to know Cmax and AUC. How, suppose 8 a.m. in the morning, the subject is given the drug, then the blood sampling will start shortly. They will take blood sample at 9 a.m., again 10 a.m., 11 a.m., 12 p.m., 4 p.m. So how to decide at what time intervals to withdraw the blood? They're not withdrawing 300 ml at once. They are withdrawing 5 ml to evaluate the blood concentrations and to plot this graph. Okay, so how to decide it whether to take first blood sample at 1 hour, 2 hour, 3 hours, 4 hours, when? So the guideline says these are the key rules. Around the Cmax, in the literature, if you know, the Cmax comes at 3 hours, suppose 3 hours the Cmax is coming, then you have to take most samples around 3 hours, because you need to capture this curve properly. So most of the samples will come around the Cmax, the 3 hours period. And the sampling should go on till at least 3 or 4 half-lives. Okay. Or better, 5 half-lives, to cover the entire elimination phase also. Okay, so the basic rules is most importantly, you should take more samples around the Cmax and keep continuing the sampling till 3 or 4 or 5 half-lives. This is according to the guideline. This will give you a proper curve, and this will give you an exact estimate of rate and extent of absorption. Taking an example of the drug, suppose a drug has a Tmax of 3 hours. Tmax means when Cmax reaches 3 hours and the half-life of 8 hours. So the blood sampling will look like this. Pre-Tmax, 0 hours, that means 8 a.m., 8:30 a.m., 9 a.m. Around 3 hours, you will see most samples. 1.5 hours, 2 hours, 2.5 hours, 3 hours. This means 3 hours means 11 a.m. Cmax is reached. 12 p.m. After you have good number of samples around Cmax, then you can reduce the sampling for 2 hours or 4 hours. Next sample will be at 6 hours, 8 hours, 10 hours, 12 hours. The frequency keeps on reducing. 4-hour gap, 8 hours gap, 36 hours, and 48 hours. Okay. Now, if the half-life is 8 hours, we can take the sampling for 5 half-lives to capture the entire elimination phase. So this 5 half-lives will be at 40 hours, but we usually take samples at 36 hours and followed by 48 hours. So you can take the last sample at 48 hours, one at 36 hours and 48 hours. It is rounded off. 24, 36, 48, 60, 72, like that, day wise. So the last sampling can be at 48 hours. So this sampling depends on Tmax and half-life. Simple. Okay. So this is sampling time points. Coming to you in the BE study design, you will find another term, housing. Now I have helped you visualize how BE study is done. Sunday evening they are called, they are served dinner. Next day 8 a.m. they are administered drug. Then they are given meals, water, sample withdrawal from time to time. Mostly these studies keep 24 hours housing. That means Tuesday 8 a.m. they will be allowed to go back home. Okay. Sometimes 2 days they will be required to stay in the clinic or this CRO. So generally housing is for 24 hours, but it can be up to 48 hours, 72 hours, depending on the half-life. So suppose in this example, we saw that the last sample is at 48 hours. That means Monday the drug was given at 8 a.m. Last sampling time point will be Wednesday 8 a.m. But the subject will not be asked to stay 2 nights till Wednesday. Okay. They can leave at 24 hours and the sample of 36 hours, that means 8 p.m. Tuesday and 48 hours, that means 8 a.m. Wednesday. They can come and give the sample on ambulatory basis. This is mobile basis. They come, give the sample and go back home. This is known as ambulatory sample. So housing is required for 24 hours. Remaining samples can be taken on ambulatory basis. Sometimes if the study last sampling time point is 72 hours, then they will be asked to stay till 48 hours. Then the 72nd hour sample they can come and give on ambulatory basis. Okay, so this is the concept of housing. How many nights they need to spend depends on the number of blood samples. Already they are spending 1 night before taking the dose, Sunday night. Tomorrow they will take the dose. Tomorrow another night they will stay. Then after 24 hours they are leaving. So that means 2 nights they are already staying. More they stay, more cost the CRO has to bear, and more cost ultimately it will be to the sponsor company. So it is always tried that housing is kept, because you have to serve the meals, you have to take care of them, take several parameters. So more the housing, more will be the cost. So it is tried to keep the housing to 24 hours in most cases, but can be 48 or 72 hours also in some cases for long half-life drugs. Okay, so this is the concept of housing. Now another important aspect, that not significantly different rate and extent of absorption means how much different is allowed. So a 20% different is allowed. So the BE criteria for Cmax and AUC is 80 to 125%. But if I say that 20% is allowed, then why it is not 80 to 120%? This is the most common topic you might have seen on LinkedIn or anything, and you may already be knowing as well. So let me help you revise that once again. Why it is not 80 to 120 and why it is 80 to 120, 80 to 125 in most cases. So actually the calculation of Cmax and AUC is the ratio between test and reference product. Suppose test product gives the Cmax of 80 units and reference products Cmax was 100 units. Then this 20% difference is not calculated. Its ratio is calculated. Ratio comes test to test divided by reference always. T by R ratio, not R by T ratio is calculated. T by R ratio is calculated. So T is 80 units, R is 100 units. The ratio is 0.8%. 0.8 converted into percentage, it becomes 80%. So this 20 variation of 20 units is allowed, and vice versa. If reference products gives less concentration, 80 units, test product gives 100 units, still the ratio is T by R. 100 divided by 80 becomes 1.25, because it's not the subtraction, it's the ratio. So 100 by 80 becomes 1.25. In percentage, it will be 125%. That's why being the comparison of ratios, it is 80 to 125%, not the subtraction. That's why the limits are 80 to 125%, not 80 to 120%. So this is how BE is concluded. The Cmax values will be finalized for test and reference products. Their ratio will be defined, and the ratio should be 80% to 125%. Same for AUC. AUC values will be found out from the graph. Their ratios will be defined and it should fall between 80 to 125%. Coming to the last topic, BCS classification. As Pranav had mentioned that we will be teaching uh, different kinds of bioweavers. So first kind of bioweaver I explained, the strength-based bioweaver, where 85 and 850 mg and 500 mg of metformin can be weaved. Only 1000 mg BE studies done. BCS-based bioweaver means you do not need to perform any BE study at all, not even on 1000 mg. That depends on BCS classification. In our third year or fourth year of BPharm, we had a subject of biopharmaceutics and pharmacokinetics. There we had a chapter of BCS. BCS is biopharmaceutical classification system. Here, all the APIs are divided into 4 classes. An API having high solubility and high permeability. That means it solubilizes in water very well and it permeates the gastrointestinal tract to go into the bloodstream. That means absorption. Permeability is proportional to absorption. So if a drug is having high solubility and high permeability, it will have no BE issues. Bioequivalent should be intrinsic property of the generic product. But if the drug is having low solubility, they will be considered class 2. Although they have high low solubility, how will it impact? If you take a tablet, it goes into your GI tract. GI tract has all this gastric fluid and all, but the API is not soluble itself. So it will take a lot of time or will not completely solubilize. Unless it is solubilized in GI tract, how will it go in the bloodstream? So such kind of products must prove bioequivalence through human studies. But those APIs who have intrinsic high solubility and high permeability, they go in GI and solubilize and disintegrate easily, dissolve easily, and go to bloodstream easily. Then generic products formulation won't impact. So BCS class 1 drugs can get a BCS-based bioweaver. Low soluble drugs class 2 cannot get BCS-based bioweaver. Class 3 means high solubility. In the GI tract, it is getting dissolved easily, but it has low permeability. So that means if you take test product or reference product, it is highly soluble. Anyhow, whether you made the tablet properly or not, the API is high soluble. So anyhow, it is going to dissolve. So for class 3 drug also, because it does not impact, the formulation does not impact the solubility, class 3 drugs also get BCS-based bioweaver. That means if you have 3 strengths, metformin 500 mg, 850 mg, 1000 mg, on none of the strengths you are required to do B, if it is either of class 1 or class 3. But if it is of class 2 or class 4, low solubility and low permeability, or low solubility and high permeability, it won't be eligible for BCS classification. So this is an important type of classification where you can save your companies 50 lakhs, 60 lakh rupees, because each BE study costs 40 lakhs, 50 lakhs, 60 lakhs. Okay, if you know that your drug is either class 1 or class 3, you can prepare an entire justification and submit to the authority in your dossier. You can get an entire bioweaver. Okay, but you have to follow certain criteria according to EU BE guidance. If your drug is BCS class 1, you have to prove in the lab that it is high solubility and complete absorption drug. It has high dissolution, gets rapidly dissolved, either more than 85% in 15 minutes or 30 minutes. Excipients between test and reference products are qualitatively same. These 3 things you have to prove in the lab. Class 3 drugs, because they have high solubility but low permeability, you have to prove more in the lab. You have to first prove the solubility, dissolution. Here you are allowed to have dissolution of 85% within 30 minutes. But for BCS class 3, it should be more than 85% in 15 minutes. only 15 minutes. 30 minutes is not allowed. And in BCS class 1, qualitative composition needs to be same. In class 3 drugs, qualitative and quantitative composition between the test product and reference product also needs to be same to qualify for the BCS-based biowaiver. So how will you know whether your drug is eligible for BCS-based biowaiver or not? How can you save money for your company? You need to search on internet, on literature, whether the drug falls in BCS class 1 or BCS class 3. If it falls in BCS class 1 or 3, then you have to perform such tests in the lab, solubility and dissolution, and compare the excipients. And if everything matches, you can propose to authority that this product does not need even one B study. So this is the BCS-based biowaiver type. Okay, so I think now let me quickly help you revise all that we covered, as the technical topics for today are over. I'll take 5 more minutes of your time. So we saw first what is a B study, why it is required to replace generics, how it is performed, blood plasma concentration profiles, B study design definitions, what is randomized, open label, single dose, 2-period, 2-sequence, crossover study, how randomization is done. We compared open label versus blinded. Open label B studies are generally open label. Blinding is not required because we are not evaluating efficacy. There are single dose studies. We cannot administer multiple doses to healthy human subjects. Then we saw how a B study is designed, how to choose the reference product. US and EU compulsorily require their own reference product. Strength selection, usually it is the highest strength where we do the B study. Then fasting or fat, usually we do fasting study, but the label, if label recommends fat study, then we do fat study. Sample size calculation depends on drug variability because pharmaceutical drugs are highly variable. So how much variable they are, based on that, you can compare them. And accordingly, you decide the number of subjects. So variability decides sample size decides BE cost. Washout period, you need to completely let period 1 treatment eliminate from the body. It takes 5 half-lives to eliminate the first drug. Sampling time points, you need to take more samples around the Cmax to have this curve properly, and 3-4 samples in the elimination phase to have the proper curve. This is the example of how you finalize the sampling time points. Housing is usually done for 24 hours because more the housing, more will be your cost. But depends, sometimes you need to do a housing of 48 or 72 hours as well. The B criteria is 80 to 125 because it's a ratio, not a difference. Then you can get entire B waiver if your drug falls in BCS class 1 or class 3. Before going to Q&A session, Pranav has requested me to introduce you to his RA Next program. Pranav from time to time runs individual customized learning program for working professionals as well as students, where according to your need, he designs different courses. If you want to learn EU market, Australia market, US market, or any specific market, or any specific dosage form, tablets, injectables, topicals, biosimilars, you can contact him and he will customize a training session for you, and accordingly he can give you a training. That entire training program is known as RA Next. And Pranav is soon going to announce his second webinar as well, which may be also another important topic like bioequivalence studies. And that will be conducted after a month or so. So you can contact him to understand what else he can provide for further training. Now, let's quickly go through the Q&A session. I will try to answer your questions as much as possible for me today. If anything is remaining, Pranav will reach out to you following. So I'll go to the Q&A session now. Somebody is asking that won't we do efficacy studies in bioequivalence study? Bioequivalence study is an indirect parameter of efficacy. It's not direct efficacy. We are not evaluating whether metformin reduces blood sugar or not. Efficacy study means we are evaluating blood sugar levels. Here we are evaluating blood concentration of the drug. So it is not direct efficacy. If the blood concentration plasma profile is good, then we assume indirectly that it is going to give good efficacy and safety. But we are not evaluating efficacy parameters as such. For atorvastatin in a BE study, we are not evaluating cholesterol or LDL, HDL profile. We are evaluating the blood concentrations. So hope this answers your question. What is the time difference between T and R in randomized study? Yeah, I think I answered it to you that it's a washout period of 5 half-lives. Third question is, if the investigator doesn't know the RLD or test after analyzing, how they differentiate it is test or RLD? See, test, if the investigator does not know or the bioanalyst does not know, they are given a sample ID number. Suppose 345 XYZ is the sample ID. Suppose there are 100 samples. All will have different sample IDs. After complete analysis is complete, then in Excel sheet, it will be revealed that this sample was test, this sample was reference, and then they will be segregated, then they will be analyzed. So blinding is till analysis is complete. Once analysis is complete, then it will be revealed, and then the biostatistician will see the differences and compare the results. Next question is, what about reference product selection for ROW countries? Very good question. For ROW countries, mostly US and EU reference products are acceptable, except for some countries. I believe Hong Kong does not accept. Sometimes they insist on doing BE study on Hong Kong reference product. So some countries may want their own reference product, but most countries, according to their guidelines you can refer, will accept US and EU reference product. Hope this answer is clear. What all criteria? Next question is, what all criteria must be fulfilled when we are filing a US approved drug product in Canada? So if you want to file a US BE study in Canada, which is approved in US already, you need to refer a Canadian guideline where you can use foreign reference product, US reference product. You have to fulfill 4 or 5 criteria. The US and Canada reference product should be qualitatively, quantitatively same. It should be IR dosage form. Resolution should be matching. Okay, so these are some whatever criteria we matched for test and reference product. Same criteria should be matched for US reference product and Canada reference product. UK ROW should not mention Australia as the preferred. So yeah, I noticed my error. It was Australia mentioned in the column, but UK accepts UK reference product. Thank you for bringing it to the notice. How exactly do we know SIVA, BV, BS question? How exactly do we know the half-life of a particular drug? Good question. This also is mentioned in the labeling. And it can also be found out in the literature, the half-life of the drug. What is the difference between, Vikas Rinde is asking, what is the difference between pilot B and pivotal B? Nice question. See, there are two types of BE studies. Pilot B study we are doing on less number of subjects, 12 subjects, because if we want to have confidence on our formulation, that we need to do a study on 36 subjects, suppose, but we don't want to risk our 36 lakh rupees. We first want to build a confidence on our formulation. So we will do a smaller study on 12 subjects. It is known as pilot study. That pilot study cannot be submitted in the dossier. Okay, pilot study is just for our confidence. If you get good results in pilot study, then you are ready to take the risk of 40 lakhs, 50 lakhs. Okay, now let's spend full money and do this. But if pilot study is not giving proper results, then you have to reformulate your product, change excipient quantities or do some other trials, and then you go for another pilot study or pivotal study. But if pilot study is giving good results, in 8 to 10 lakh rupees, you are able to know that it is giving good results. Now you are ready to spend 40 lakh rupees. So pilot study is not for dossier submission. It is just for confidence. Pivotal study is the one that we discussed today. And according to the guideline, pivotal study should be submitted in the dossier. Siva BVBS is asking, in reflection paper of dissolution, it was given that Q point is taken from biostudy data, but as per my knowledge, the Q point is decided by the R&D team, right? Based on trial and errors basis. I think, Siva, this, according to reflection paper, it is correct. The Q point has to be taken from biostudy results only. If in your biostudy batch, whichever batch you used, if it is giving 95% dissolution, then you have to keep 85% Q. R&D will not decide. Bio batch results will decide whether it is 85% Q, 80% Q. Okay. So if it is not decided according to reflection paper, authority will ask you a question and they will not accept until you comply to the reflection paper. It is one of the most frequently asked questions. Thank you, Manali, for the appreciation. To do if pilot and pivotal both B studies are failing. If pilot or pivotal B studies are failing, then you have to reformulate your product or either look for BCS-based biowaiver. Reformulate the product, again, make it better and do the pilot and pivotal B study again. Any specific guidelines for new polymorph drugs? I think it is not in the scope of today's discussion, so we will skip this question. Last question comes from Abhijit. B study for fixed dose combination drugs. Yeah, I think it is also a little out of scope, Abhijit, but for fixed dose combination drugs, you need to do multiple B studies. Okay, it depends whether both the APIs or 3 APIs are in a monolayer or 2 layers. In the EUB guidelines, it is clearly explained whether you need to do 1 B study, 2 B studies, or more B studies. How quantitative proportionality you will evaluate. Okay, if it is a double layer tablet where each layer has a different API, then it becomes easier to prove proportionality. But if it is a monolayer, both the APIs are in the single layer of the tablet, then it becomes difficult to justify the quantitative proportionality, and then you have to do more than 1 B study. I recommend you go through EUB guideline for immediate release products. There it is clearly mentioned, 1 or 2 paragraphs, how you perform FDC products B studies. So I think all the queries are now resolved. Even if you have further queries, you may reach out to Pranav. He will get back to me and I will answer your queries. So thank you everyone for your time and attention and patience. Hope to see you again. Thank you. Goodbye.
Thank you all. If you have any queries, you can mail me. We will try to answer you.
Okay. Goodbye.